I'd like to thank Bennett and Tom for inviting me back to talk with you today about in this symposium. This is a very huge interest of mine, and I'll tell you a little bit more later. But first I want to talk about this beautiful scanning EM that was taken by my lab manager, Yu-Ying Liu. And this shows exactly what Carol talked about, is a cell that has mesenchymal characteristics. In this case, this is an ovarian cancer cell invading into a layer of normal peritoneal mesothelial cells. So this process that she discussed is very relevant in tumor biology as well. And so just a little bit of background. What I want to do is to go into a little bit more detail about some of the specific molecules that Carol already talked about in the EMT context, and not bone-crushing amounts of detail, but just again to give you a general picture. And I can really appreciate, I think, you as an audience. I don't know what your background is, but I'm assuming it's not cancer biology. I came to Northwestern in 1994, so a little bit ago, as a biochemist and mechanistic enzymologist. And during my time here, I was sort of pounded into a tumor cell biologist. And that transition I thought was really painful at the time, but I had absolutely no clue. I've spent the last couple of years having a lot of conversations with engineers and physicists and mathematicians, and those conversations are way more painful. so forget about the chemist. So those conversations start with me going into somebody's office, and I say, so what do you do? And they say, microfluidics. Something. I have no idea what they're talking about for about a half an hour. And then a word comes, literally, I'm serious. So then a word pops out that I can relate to, like a simple word like separation. Oh, okay, so a microfluidic can be used to separate stuff. Okay, now I know what you're talking about. So you separate on the basis of size or charge or chemical properties, all of the above. Then we can talk about, okay, can you use it to separate this from that, and tumor cells that have this microRNA or whatever. So those dialogues are initially really painful, but they're really important, and so I really encourage you to have them. And to somebody like me, that's actually really interesting because I'm tired of hearing myself talk. So I've been working on the same things for, since the early 90s. And those, so I can pat myself on the back and say I'm really smart when I'm talking about the things that I know a lot about. But when I talk to an engineer or a physicist or a mathematician, and sometimes even a chemist, I can't answer any of the questions that I'm asked, and that's a good thing. You shouldn't always be able to answer all the questions that you get, because then you kind of get complacent and stop thinking. So I'll get off my soapbox a little bit and talk to you about some ugly statistics. So these are the 2010 statistics, but they haven't gotten any better in the last year and a half. In the US, every year, there's about 750,000 men and 750,000 women that are diagnosed with cancer every year. And this slide shows the cancer deaths. So of those 750,000 that are diagnosed, about 40% of them will die. So it's very important to think about this as a very major health problem, and this is only in the US, not to mention the rest of the world. So cancer sucks. I think we can all agree on that as a basic premise. And the other thing is that probably every one of you has a friend or a family member or a relative that's been affected by cancer, so it's something that everybody can relate to. And I think that we definitely need some new approaches, and I really applaud this Physical Sciences Oncology Center concept. Even though the conversations are initially difficult, they're really important to have. So I started looking at what I wanted to talk about. And some of the questions that I think about a lot are, why is metastasis important? What is it to begin with? How do we study it? And how do we stop it? And I had asked Jennifer and Munchie what they were going to talk about. So they're going to talk about the last two. So that left me off the hook for two real big topics. So I won't talk about those at all. I should have touched base with Carol, too. So why is metastasis important? That's a really easy one to answer because it kills you. And the majority of people that have cancer don't die from their primary tumor. They die from the metastasis. So understanding factors that initiate metastasis, that promote progression of metastasis, that stop metastasis, that allow us to image metastases, to treat them, are of vital importance, because that's what's going to kill those 40% of men and 40% of women with cancer. So what is metastasis? It came from a Greek word, as a lot of cool words do, that means next placement. And it's simply, as Carol already alluded to, the spread of a tumor from the primary site to somewhere else. Again, when tumors are at the primary site, and if they're just growing there, you will eventually find them. And if all it took was to cut it out, you'd be fine. But it's the metastases, the micrometastases, the stem cell population that Carol alluded to that are detrimental. And this commonly occurs through either blood or lymph, with some notable examples like the ovarian cancer cells that I just showed you. So this is just a picture illustrating that concept, and I took all of these off of Google Images, and most of them I forgot to put the website, and I apologize because I don't usually do that. So these are not my drawings, but the primary tumor is shown here, and the metastasizing cells that break off of the primary tumor are shown here, entering the vasculature. As Carol alluded to, tumors are angiogenic. They're very hungry because they have all these cells that are dividing like crazy, so they need more food. They basically induce blood vessels to grow into the tumor mass, and then they use that as a conduit to go elsewhere. This is another picture of it, showing it in a little bit more molecular detail. What the tumor cells from the primary tumor, again, is going to be up here. And the factors I want to focus on in a little bit more detail are here. So the cells will attach to the blood vessel. They'll break down those cell-cell junctions in the vasculature and invade and migrate into the vessel, float around somewhere, and eventually repeat the process in reverse, essentially, at the secondary site and form a metastatic lesion. So the process of EMT that Carol was talking about is very active in this particular transition. So I'm going to talk to you about a couple of factors or molecular requirements that are really needed for cells to successfully metastasize. And each of these factors is a gigantic field unto itself, so I'm really not going to give you a huge amount of details, but really just introduce you to the basic concepts. So think of me telling, somebody telling me what microfluidics is in a way that it separates stuff. So I'm giving you that level of explanation. So there's obviously a lot missing. But we're going to talk about adhesion receptors. That could be cell-cell or cell matrix, a little bit about proteases that are going to remove the physical barriers, and motility and invasion itself. So this is a good way, I think, to think about adhesion receptors. You've got a plate of sticky buns, and if you want to have one, it's stuck to its neighboring sticky buns, so it's stuck here, but it's also stuck to the plate. And the tumor cells are like sticky buns that you want to take one away, and you want to go somewhere else with it so that you can eat it in private. But there's a lot of things that are sticking them to each other and to the plate. Adhesion molecules are different also than sticky buns, so that the analogy ends somewhere, but they also provide input on the tissue context. So the adhesion molecules are very important in development and in tumor biology and telling the cell where they are in the organism. Do I have neighbors? Are they going to let me spread out? Can I really sprawl my stuff all over the row, or do I have to sit like this? Can I move to a different spot? They can be homophilic or heterophilic, as the name implies. They stick to themselves or stick to other types of adhesion molecules and are important in development as well as disease. So there's a lot of different types of adhesion receptors. The two types that we study most frequently in cancer biology are in the cadherin family and the integrin family. And again, Carol alluded to these already in her talk, and I'll just talk to you a little bit more detail about those. So cadherins participate in cell-cell adhesion. So these are the molecules that stick the sticky buns to each other. And so what we're looking at here is this is the membrane of one cell. This is the membrane of another cell. Cadherins stick out into the intracellular space, and they bind the cells together. There's a lot of different types of cadherins, depending on what types of tissue expresses them. So epithelial or E-cadherin is probably the most famous. There's cadherins on endothelial cells and on neural cells as well. And another very important factor in these adhesion molecule families is that they link the cell-cell adhesive process with the cytoskeleton. The cytoskeleton, as its name implies, is the skeleton of the cell. It maintains its structure. It helps it move around. It moves organelles back and forth where they need to go. So adhesion molecules are essentially important for basically transducing information from the outside of the cell into the inside and telling the cell what to do. Is there another epithelial cell out here, or can I move? Am I stuck here? Do I need to go somewhere else? This type of information is transduced through cadherins. There are two types of cadherins, classical cadherins like the ones I just described to you, and desmosomal cadherins like the ones shown here. And these are essentially serve the same function. They stick one cell to another cell, and they're linked to the cytoskeleton. A major difference is that the classical cadherins are linked to the actin-based cytoskeleton, and the desmosomal cadherins are linked to the intermediate filament cytoskeleton. So they link to different parts of the skeleton, to tells the cells different things to do. But again, complicated molecules. These are heterodimers, so there's a molecule of desmogliin and desmocolin linking to another dimer on another cell, whereas E-cadherin are homodimers. So what do they look like? Well, you've sort of seen some of them in Carol's talk, but I'll show you a few more. Are not necessarily tumor structures, but in the uterus, there's a layer of epithelial cells, and that's here on the top, and the red part is showing E-cadherin. So again, where the cells are interacting with each other, you can see nice E-cadherin staining. Similarly in skin, this one looks a little bit blurry, but you can see in the epidermis, there's very nice staining in brown for the E-cadherin on the top of the skin. In tumors also, so this is a ductal carcinoma in situ. This is an early stage lesion. It has not undergone EMT yet. And you can see that it maintains E-cadherin staining, the brown stain, so each of these is a cell. This is the normal breast tissue out here. And again, in a lung cancer, again, these are very well circumscribed tumors. They're not metastatic at this point. They maintain E-cadherin expression, and they're not really going crazy going into that stromal tissue. So this is a desmosomal cadherin with similar staining. Okay, the other type of adhesion receptor that's very important are integrins. So this is the part that sticks the sticky buns to the plate instead of to the other buns. And these are expressed in a wide variety of cells. Again, they're transmembrane molecules, so this is the outside of the cell. This is the inside of the cell. And they have an alpha-beta structure, so every integrin is going to be called alpha something, beta something, like alpha V beta 3 or alpha 2 beta 1, something like that. And what they do is to bind the cells to the extracellular matrix, not to other cells, but to matrix proteins outside of the cell. And these are very important in motility and invasion, as I'll show you in a couple of minutes. And this gives a little bit more detail on some of the structures. Again, I think we don't need to get into that level of molecular detail because I could talk to you for about 3 hours on that. But if you're super interested in that, I'm sure that there's multiple graduate courses that you could sit in on. I really like this picture. I wish I could take credit for drawing it, but it shows, I'm not a good three-dimensional thinker, so pictures like these are very helpful to me. Showing the integrin, this is the cell membrane, this is the junk on the inside, this is the stuff on the outside. So the extracellular matrix is a meshwork of lots of different proteins interacting with each other. For example, collagen. Collagen is the major matrix protein in the body. About 25% of your body is comprised of collagen. So you really want to keep it in the right orientation. You want your cells to stick to it so you don't turn into a slime mold. And there are other matrix proteins like fibronectin, laminin, some proteoglycans, et cetera, in here. And you can see that both the alpha and the beta subunit of the integrin are important in binding to that matrix protein. Now, there's a huge amount of stuff going on down here, and I'm just going to show you some very generic diagrams of that. The cytoplasmic face of the integrin also links to a very rich network of cytoskeletal and signaling molecules. So like the cadherins, it tells the cell what's going on the outside. Is my matrix intact? Is it stiff? Integrins are very well known to be mechanotransducers. So one thing that happens in tumors, for example, is that they become fibrotic. And what that means is that the tumors, for some reason, induce the formation of a lot of extracellular matrix. And there have been some very interesting studies done fairly recently showing that, actually some of them were done in Munchie's lab. I don't know if he'll be talking about that later or not, but showing that the matrix stiffness is important in transducing differential signals to the cell. And those initial studies in tumor biology required a collaboration of cancer biologists with engineers to do the nanoindentation and the AFM that was required to take those initial stiffness measurements on human tumors so that the tumor biologists could then go model it more appropriately in the lab. So integrins will detect the matrix structure, the matrix composition, and the matrix stiffness and transduce that information into the inside of the cell. There's a lot of integrin heterodimers. It's very complicated. There's no good rule saying alpha-2 beta-1 is always upregulated in cancer. It totally depends on what type of cancer you're looking at, what stage it is. It might not be upregulated or downregulated at all. It might just be functioning differently. It might be interacting with different stuff inside the cell. So there's really no good rules about that. But the one point I want to point out from this slide is that some integrins are promiscuous and some aren't. So for example, alpha-6 beta-1 mainly binds to laminin, whereas alpha-V beta-3 can bind to multiple matrix proteins. And this is just another diagram showing essentially the same thing. So laminins can bind to lots of different matrix proteins. Integrins can bind to lots of different matrix proteins. Some integrins can bind more than one protein. Some proteins are bound by more than one integrin. Integrins signal, I alluded to that. So at the cytoplasmic face of the integrin, there are a lot of scaffolding molecules that aren't shown here, and these scaffolding molecules link the integrin to all of these various types of signaling molecules. And again, which molecules they link to depends on the matrix structure, the matrix composition, and the matrix stiffness. And depending on which signaling pathway that they link to, it's going to tell the cell to stay alive, to start dividing, get out of here. You should be a duct, something like that. So the matrix signals to the integrin through these variety of pathways. And as we know, I've got to stop saying pathways, they're actually networks. It's not a linear fact goes to differentiation, but they're actually joined in networks that are regulated by multiple things. And I don't have a slide illustrating it, but there's actually molecular crosstalk between the pathways that are activated by integrin engagement and those that are activated by cadherin engagement. So a cell needs to know not only if it's undergone an EMT and it's a migratory cell off on its own, then there's no cadherin signaling going on, but if it's going as a cohort of cells during development or during metastasis, the signals from the adjacent cells are as important as the signals from the matrix. It's complicated, as Carol showed. This is the adhesome now. This is one integrin interacting with a gajillion other things, which is why it's very difficult to come up with any hard and fast rules about integrin signaling or integrin interactions. But just to know that there's a lot of levels of regulation, and that also means then there's a lot of places where the regulation can be screwed up in tumor biology or other diseases. And conversely, then there's a lot of potential drug targets in there as well. So what do integrin adhesions look like? I think I just have one picture of this. So this is a fibroblast. The rest of the cell is out here where you can't see it. And these are the filopodia that are coming off of the migrating cell. The green is actin, and the red is vinculin, which is one of those scaffolding proteins that associates with integrins. So you can see the integrin binding to its substratum here, binding the rest of the cell. If you look in tissues like skin, again, there's a lot of integrin staining in the skin. Looks very similar to what we saw for cadherin staining. There's a lot of matrix proteins here and matrix proteins there that it's binding to in the tissue. And I just like this because it was pretty. It doesn't really have that much to do with anything. This monitor just went off, if that matters, but I can look up here. And what this is a micropattern matrix, and somebody probably put this out around the holidays, I would imagine. And you can see actin and tubulin staining and nuclei, but it makes a very nice image. And I know there are groups here that do that, and I want to play with you because I want a Christmas card like that. So proteases are the next topic I want to talk to you about. And what proteases essentially do are remove physical barriers to migration. And I thought that this was an appropriate image for that. Maybe I've been at Notre Dame too long already. They are, so this guy here could be viewed as a key protease, removing physical barriers to migration. This can be done by modifying the matrix or also by modifying cell-cell junctions and processing cell surface proteins and growth factors, et cetera. There's a lot of different kinds of proteases, again, like integrins. There's a gajillion of them. I'm not going to get into all the details, but there are multiple molecular classes of proteases based on what type of chemistry that they use to cleave the peptide bond. The ones I've focused on most in my lab are metalloproteases and serine proteases. And again, metalloproteases generally use zinc to polarize a water molecule to initiate peptide bond cleavage. Serine proteases use a serine hydroxyl group to do the same thing. This is an example of my current favorite protease. This is called membrane type 1 matrix metalloprotease, or MT1-MMP, or MMP14. Metalloprotease, it has a zinc in the active site, which obviously isn't shown here in this generic structure. What I wanted to illustrate here is that some proteases are linked to the cell, so this is meant to be the cell membrane, the rest of the cell body's here. So these proteases are in the extracellular space. Some proteases are secreted into the extracellular space and are active in solution phase. Proteases like MT1-MMP are active in substrate cleavage. This is a very important protease because it's a type 1 collagenase. As I mentioned, 25% of mammalian organisms is collagen. So collagen-lytic enzymes are extremely important. Regulating their activity and basically keeping them off is extremely important, so you don't have too much collagen degradation. You get a lot of pathologies, not only invasion and metastasis, but things like arthritis and other connective tissue diseases. So these proteases can directly cleave substrates, but they can also participate in zymogen activation. And what that means is that this active form of the protease can activate an inactive form of a second protease. In this case, MMP2, or gelatinase A, as it used to be called. And the important thing about this is that if you get a few proteases like this in what's called a zymogen activation cascade, one molecule of MT1 will activate 5 MMP2s that'll activate 20 of some other enzyme. So you can really get a cascade effect to amplify the proteolytic problem in the case of cancer biology. And for targeting purposes, then it's more efficient to target the initiator of the zymogen activation cascade than one of the downstream proteases. There's some super interesting biochemistry involved in this enzyme as well, but that's a story for another day. So this is an example. This is a cell, most of a cell, the rest of it's back here. You can see the moving edge, the leading edge here. And the MT1-MMP is expressed on the outer edges of the cell. This is showing the three-dimensional collagen matrix that the cell is moving through. And here you can see that the cell is interacting with the three-dimensional collagen, invading through that three-dimensional collagen matrix, such as you might see in the skin, in the stroma of a breast tumor, et cetera. And I'd like, when I use this slide when I'm teaching enzymology, to say that everybody's heard E plus S goes to ES complex, goes to E plus P. I don't know if you guys have that or not. But this is a nice picture to show the enzyme substrate complex. It's better than kind of the E plus S or the sort of complicated chem drawings. Okay, so another thing that proteases do, so we always thought of them for years and years as something that's going to be on the outside of the cell or very near to it and catalyzing movement through the matrix during development or tumor progression. But the other thing to do is, like you guys down there, you can't really spread out because they're in your way. So if you were a protease, you would need to get them out of the way so that you could spread out and have room to grow and divide. So proteases are also important for removing physical constraints within the matrix that stop proliferation. So this is, in this assay, what we did here was to take ovarian cancer cells and seed them into one cell into a dense collagen matrix. If they don't express MT1-MMP, they divide a little bit. This is the same magnification as this. I just cut off the rest of the gray. But if they express MT1-MMP, they can really grow into these very large multicellular aggregates with invasive projections, and they can really spread out and start to invade. So another way to think of tumor-associated proteolysis is removing those physical constraints that are imposed by that fibrotic matrix on the cells so that they can spread out their cytoskeleton and undergo effective proliferation. And oh, that one looks like Satan. I had that up there before I moved to Notre Dame. So but there is a little bit of a theme here. So the last thing I want to talk about is just a touch on motility and invasion. And this is the study of motility and invasion often centers around the cytoskeletal changes that enable cell movement. And to me, this is a lot more complicated than the previous two. I don't know why, but it's harder to digest than than the other concepts to me. So I think that the two things to think of is that it's precisely controlled like clockwork, and it results in invasion like these aliens here. So it requires a lot of concerted steps that I'll show you in diagrammatic form in the next slide. But first, the cell has to protrude projections in the direction of motility. It requires new adhesive contacts that can require proteolysis. The cell has to let go at the back if it wants to move forward. There has to be contraction like a muscle on a much smaller scale. And there are lots of signaling factors that are involved in that. And very, very generically, there's a family of enzymes called Rho-GTPases that are involved in various steps of motility. And these are the three that you're most likely to hear about. Rho is involved in formation of stress fibers, RAC in formation of lamellipodia, which I'll show you what that is in a second, and CDC42 in filopodia. But these events have to be finely orchestrated and coordinated, well coordinated with each other. So this is a diagrammatic form of what I just showed you. So if this cell wants to go that way, for example, if a tumor is secreting an angiogenic factor, and this is an endothelial cell, moving toward the tumor, or other examples. So this requires the cells, it's migrating on a matrix. So this is important too, that extracellular matrix is really important for migration. And the way to think about that is it's a lot easier to walk on grass than it is to walk on ice, because if your feet can't stick to something a little bit, you really can't go anywhere. So you have to have adhesion to have motility. That's a little bit counterintuitive, but you have to have a little bit of adhesion to have motility. So the cells will extrude a filopodium in this case, or a lamellipodium. Maybe I don't have a picture of that, but that's a more larger type of projection. Adhere to the underlying matrix. This requires the whole cytoskeleton to reorganize, and that's where those Rho-GTPases are critically important, whether it's organizing a filopodium, a lamellipodium, the whole stress fiber network, et cetera. The cell is under tension, so there's a mechanical signal that's involved here as well for the cell to move forward. This requires deadhesion at the leading edge. The integrins in many cases are ripped from the plane of the membrane and deposited in the matrix, which I find to be weird and inefficient, but it appears to be true. And this will then lead the cell to move in a given direction. And again, I'm not going to go through this. This again shows that there's a lot of networks, and there's a lot of things polymerizing and depolymerizing in the cytoskeleton. Integrins are involved here, and all the integrin-associated contacts, a lot of cytoskeletal things again, too much detail to be particularly useful, but just to know that this is a lamellipodium. I knew I had one somewhere. In order for this leading edge of the cell to go forward, it requires a lot of very coordinated cellular processes. So again, this is an example of collective migration. Now you have a protease here at the front and the cells trailing behind. And this is an example from a tumor spheroid moving through a three-dimensional matrix. And you can see here that the cells are moving in a collective fashion, as Carol alluded to also in development. And this is, the research in this area is very much ongoing, but it's believed to result from limited proteolysis creating very fine-scale defects within the matrix structures that kind of forms a track that other cells can move along, and other things that are deposited in the matrix. So that these cells stay, they're not stained for E-cadherin, but these cells stay connected to each other and migrate as a collective. And you see this a lot in development, and you can see it in some very specific types of tumors as well. So this is why Is Metastasis is important. This is what happens when all that works properly. You die, essentially. So this is a woman with end-stage ovarian cancer. You can see huge amounts of intraperitoneal metastasis, and you will die. So that's why it's important to study metastasis. So the way we've done metastasis research in the past, and certainly when I came to Northwestern, and it's slow to change now, is we each have our favorite flavor of jelly beans, and these are, can be representing various disciplines. And we like our jelly beans, and we talk to our, orange jelly beans, talk to orange ones, and yellow ones talk to yellow ones. But that doesn't work in most cases. There have been some really great strides in metastasis research, but for the most part, there wouldn't be 40-some% of people that get cancer dying of metastatic disease if this approach was working. So we need to do this, and I think that's why we're here in this room in this type of physical sciences oncology center. We need to mix up our jelly beans and take a whole handful at once, and they actually are delicious like that, even though at first it's kind of weird. And this is an example of some of the jelly bean handfuls that I've been eating lately, and I don't pretend to understand a lot of this, so microfluidics was the example I gave early on. We're working on a collaboration to, there's an increase in oral cancer in the population of the age of most of this audience, except the gray-haired section down here, that is associated with HPV. So traditionally oral cancer affects over 60 men, heavy smoker, heavy drinkers. There's a huge increase, 225% increase in the United States in oral cancer associated with human papillomavirus, the same ones that cause cervical cancer. And that's affecting the 20 to 40 age group. So it's going to be a huge public health problem in your lifetime. It's difficult to detect. Most oral cancers are detected in the dentist office. So we're collaborating with a colleague in engineering to try to use a microfluidic device to detect HPV in an oral rinse sample. And if successful, this could be something that could be done in the dentist office as an early detection mechanism for just a gargle, spit in the cup, and then they put it in the device. And if it's positive, you go to the next step to an oncologist. I showed you the example of the patient with late-stage ovarian cancer. So what you saw there is multiple intraperitoneal metastases. This is a different type of metastatic mechanism than what I just described to you, and I won't go into the details. But one thing that's very typical of ovarian cancer is that these women develop a lot of fluid in the peritoneal cavity. Normally, the fluid in the peritoneal cavity is about 5 mils. The intraperitoneal pressure is actually subatmospheric. And in women with ovarian cancer can develop up to 5 liters, in some cases more, of ascites fluid. And this increases, this does a lot of things, but one of the things it's doing is increasing the intraperitoneal pressure. And we're trying to ask the question, and I have no idea how to do this, so we're just starting to work with engineers and some computational folks as well to try to figure out a way to address this question. Does that increase in pressure in and of itself affect either the competence of the cells to make them interact with their signaling molecules to make them more effective at metastasizing, or does it potentially also interact with the peritoneal cavity and make it more receptive to being implanted? But that's, if those of you who are engineers, if you think of 5 liters of fluid in, say it's a sphere, even though it's not, you have a compressive force, you have non-laminar flow, you have strain as the balloon stretches, and so from a cancer biology point of view, that's where do we start sort of problem. So we've started a collaboration here to use an instron. Basically, we grow cells in fluid-filled bags and put them in the stainless steel canister and put some pressure on them, and are trying to figure out what's going on. Cold atmospheric plasma, I won't go into a lot of detail because we've got more speakers coming up, but cold atmospheric plasma may be useful for selective killing of skin tumor cells. Laser transmission spectroscopy might be used to detect cancer antigens, and we're trying this with prostate-specific antigen as a proof of concept example. Live cell lithography to recreate a metastatic niche. Bisphosphonate functionalized nanoparticles to detect bone metastasis. Statistical modeling of large-scale data sets that are publicly available to detect cancer relapse. And another microfluidic example to look at single-cell proteomics. So I think that these innovations are really what we should be encouraging, and I would encourage you, even though it's a lot harder here, to get down to Chicago and talk to your colleagues there. Maybe, I don't know if you can video conference or something like that, but it's totally worth the conversation, even though it's painful at first. I think that there's a lot of interesting and innovative things that can come of it, and you'll get asked a lot of questions that you can't answer, and that will make you smarter. So I'd like to thank our funding sources, the National Cancer Institute, the Walther Foundation, the University of Notre Dame, and the Harper Cancer Research Institute. And I'd also like to challenge those of you who are true dyed-in-the-wool geeks to spell your own name out of actual data. We do this every year now, and it's fun in a dorky sort of way. So I'll stop here and answer any questions that you might have. Well, if there's no questions, we can just move on. And oh, there's a break now? Okay. The computers stopped working too.
Hi. How are you?
Are you Mike?
Yes, yeah.
Munshi.
So I have your slides on the desktop here.
Perfect. All right.
Did you want to look at them?
That's fine. As long as I know. As long as you check them out.
Okay.
Right?
Yeah.
Perfect.
Okay.
And in terms of what you have here, this is a light.
Yeah.
Where's the light part of it?
It should be. I think you just press that part.
Let's see.
Oh, here it is. Right in the middle there.
That's the light. And then to move slides.
Yeah.
I see. All right. Perfect. Good. Thank you.
So the,
With the hook you
um,
have, yeah.
I think you're gonna have to talk in here because we had the mic, but it was having some technical difficulties.
All right.
Is that gonna be okay? The tech guy should be coming back
Yeah,
soon.
I prefer to normally I prefer to stand somewhere here.
Yeah. Okay.
See what he can do.
Okay. I'll see what he.
You can use a handheld.